My name is Anika Correll Trnka. I'm working with ESR. My current project is looking at attributing mixed body fluids to individual donors. When you have a crime scene stain, you can do like conventional body fluid identification tests to figure out what body fluids are in that stain. So you might have a mixed stain of saliva and blood, and the test will tell you that. You can also do your usual DNA profiling stuff to figure out who may have contributed to that stain. So you know, person A, person B. But those body fluid identification tests and the DNA profiling are independent tests. It doesn't tell you which body fluid came from which person. You can imagine that could be pretty important in a case, you know? Often these days, the DNA match itself isn't what's in question, it's where that DNA came from. So I'm looking at using mRNA sequences to attribute those body fluids back to their donors. And I'm specifically looking at C-SNPs, which are single nucleotide polymorphisms in the coding regions of DNA. And these are just single base pairs in the DNA that differ between individuals. These single base pairs that differ are copied into the mRNA sequences. So then we can use mRNA sequences to distinguish between people, just like we use DNA sequences to distinguish between people. The other feature of mRNA that makes it so useful is that it's different between different body fluids. If we put that all together, you can see we can extract the RNA from a stain, we can figure out what body fluid it came from, and we can match that back to the individual. And if the SNPs match, we can say, well, that body fluid may have came from that individual. It's a very complicated process, and gene expression is not as straightforward as we would always like. Sometimes some SNPs don't show up in the sample, even though we know that their DNA has that SNP. It might not show up in the RNA because of preferential expression of one transcript over another. So if you have two copies of your DNA, one from your mother, one from your father, and you might have two different SNPs. Your DNA contains the two different SNPs. On the RNA sequence, we only see one because one of them is being expressed rather than the other. There's also issues with genomic DNA contamination in your RNA. So you extract the RNA from the sample, but if there's any DNA left over, that can introduce spurious alleles that we wouldn't otherwise expect to see. So we need to develop the methodology to circumvent those issues like genomic DNA contamination, and also develop the statistical techniques to deal with biological phenomena such as that preferential expression. There's definitely a lot more to be done. We can look at evaluating a wider range of C-SNPs and mRNA markers to use. And then hopefully one day we can extend this even to proteomics. So looking at the proteins, which can also differ between individuals, and using those for donor attribution.